Okay, the final lecture in the series of cellular respiration is the oxidative phosphorylation pathway. So remember the big ideas that we're trying to get out of this of what goes into this reaction, what comes out, and what are the energy related molecules involved in this process. So as you can see, oxidative phosphorylation involves two parts. We'll talk about the electron transport chain and chemiosmosis. And as you can see from this overall diagram of cellular respiration, we are inside the mitochondria. We are using electrons that are being transported with the NADH and FADH2 molecules. They're coming into oxidative phosphorylation as well as products from glycolysis, and ultimately through the complete oxidation of the glucose, we end up with ATP. So this is a picture that shows you that the oxidative phosphorylation pathway happens inside the mitochondria on that folded membrane called cristae. And so this picture here is representing not the outer membrane of the cell, but the inner membrane of the mitochondria. And this folded structure of the cristae is very important because it increases the surface area and the number of these components, which are proteins, and they're going to move the electron from one location to another, one state of energy to the other, and ultimately we will end up with some ATP. So all of the NADH and FADH2 molecules that have been previously made in the previous steps of cellular respiration will be moved through a series of oxidation reactions in the sites of these proteins, and their energy will be harnessed into ATP. So this again, I'm just showing you a different picture of the molecules. I've circled up here showing that incoming are NADH molecules and FADH2 molecules that comes both from the citric acid cycle and from glycolysis. And this is the actual pathway where most ATP is formed from the oxidation of a single glucose molecule. So there's two parts, as I mentioned. There's the electron transport chain, and the electron transport chain is actually just a series of protein molecules that are found embedded in the cristae, that inner membrane. And as the products of the glycolysis and citric acid cycle are fed into the electron transport chain, you're going to watch a series of reduction and oxidation reactions occurring. They've numbered the participants in the electron transport, these proteins, with Roman numerals 1 through 4, and we are going to oxidize the NADH and FADH2 molecules, and the electrons that come off of those will be passed through these complex of proteins, going from a high energy state to a lower energy state. There is no ATP, as you can see if you follow these arrows along. There is no ATP actually created. There is simply the passing of electrons and changing of energy states. So ultimately what happens is as the electrons, which came off from oxidizing NADH and FADH2 molecules, they will ultimately be picked up by a molecule of oxygen, and you can see that here, and that is created into a water molecule. So this is just showing you another picture of this happening. So you can see that the electron is being passed from one to another, and ultimately what happens to the electron is picked up by oxygen and created into a water molecule. The second part that you're seeing over here, it leads us to chemiosmosis. And chemiosmosis is called the energy coupling mechanism. And this is also occurring, same location, inner membrane of the mitochondria. And we're going to use a different protein that's embedded. And this is an enzyme complex called ATP synthase. So that's what this big blue thing is. And this ATP synthase actually works like an ion pump that's in reverse. So instead of pumping ions from low to high, it actually allows the flow of ions through it from high to low and generates the energy from that potential energy movement, high to low, to put together ADP and an inorganic phosphate. But remember that this is in reverse. So it's not actually doing active transport. It's using the flow from high to low to create this ATP, and that's actually called chemiosmosis. So this shows you another picture of, we have a high concentration of hydrogen ions on the outside of the membrane, and on the inside of the membrane we have a low. And as they flow down their concentration gradient, you can see that this actual ATP synthase protein will actually spin, and that spinning, that energy collection, is what forms the bond between the ADP and the inorganic phosphate, thus creating ATP. So one thing that you've noticed from both these processes, electron transport and chemiosmosis, is that all the reactants and all the products were energy related. We had NADH and FADH2 coming in. In fact, there were 10 NADH molecules. I have a typo here. That should not be a 2. Sorry, I will fix that. And 2 FADH2 molecules. And those are all coming in from glycolysis in the citric acid cycle. At the end of oxidative phosphorylation, both steps, chemiosmosis and electron transport, we have generated approximately 34 ATP molecules. One NADH fed into the system can generate about 3, and FADH is a little lower energy molecule, so only 2 ATPs can be generated from one of those. So make sure, I've double kerwhammied this slide, make sure you understand all of the whole process of cellular respiration from beginning to end, that under optimal conditions, glycolysis will give us 2 ATPs. That prep reaction where we change it to acetyl-CoA, we don't make any ATPs from that. We make 2 ATPs out of the citric acid cycle. So at this point, we're at a total of 4. And then when we bring oxygen into the equation, which is the oxidative phosphorylation that happens in the cristae, we can come up with 34 net ATPs. So if you add the anaerobic, which is up here, the 4, to the aerobic, the 34, we say from one glucose, when you run it under optimal conditions through the cellular respiration metabolic pathway, 38 ATPs are created. Now, is glucose the only thing that can run through the pathway? As I've mentioned before, the answer is no. As you can see, we can feed proteins, we can feed fats through the process, but they have a different pathway to go into the different parts of cellular respiration. The pathway, however, is less efficient and for proteins and fats, and a little slower. So even though fat supplies us with more, it's a higher potential energy molecule than glucose. As mentioned here, the pathway just isn't as efficient to break that down as glucose is. Now, what about those organisms that do not use oxygen, or even within our bodies? What if we're at an oxygen debt? The process of fermentation is defined as the anaerobic breakdown of glucose. And if you can see as you can see here, okay, we end up with 2 ATPs and a toxic end product. And that toxic end product we're going to get is called lactic acid or lactate. And that is from changing pyruvic acid by addition of 2 hydrogens from NADHs, 2 of those, into this still 3-carbon molecule, but it's gained 2 hydrogens, which is why it turns into an acid. All right. This process of NAD plus production is what actually can feed into glycolysis, and this can keep going over and over and over. So this shows you the difference between fermentation and glycolysis. We don't usually say our cells ferment. Can we, when we make our cells in a condition of oxygen and debt, instead of following this pathway, follow this pathway? Yes. But as you can see, we can feed back into the glycolysis pathway those NAD plus molecules. So it's a way for our cells to quickly regenerate electron acceptors, NAD plus molecules, so we can keep this energy pathway going. So this shows you the pathway again of lactic acid. This is called lactic acid fermentation. Here is your lactate or lactic acid. And that lactate is toxic and will build up in your muscles. So this is probably what you've maybe heard of as an oxygen or a burning sensation in the muscles. Now, I've actually read some studies that they're not sure if it's exactly the lactate. We've always associated with that. But what you should know is that this lactate will build up in your muscles when you put your muscles in an oxygen deficit. So when you're trying to work your muscles too fast, too hard, and you can't supply them with oxygen fast enough to do the aerobic portion, oxidative phosphorylation, they will follow this quick, easy pathway. And the only way to get the lactate out of the muscles is to supply with more oxygen, which typically this lactate is carried via your bloodstream to your liver, where it's converted back into pyruvate, which can be fed through glycolysis. And that is it for cellular respiration. Make sure you email me any questions, and I will see you on Tuesday.